Hello, y'all. This is Benny Chen here for an introduction for Miss Richardson's 4B class, Group 4 project, the Ecocolumn. So, as you can see on the other pictures, this is what an Ecocolumn looks like, but this is a modified form for how we have it. So, as you can see, in the Ecocolumns, it's a habitable for both organisms, the aquatic organisms and earth organisms. So, first, we have the top layer, the soil with worms and plants living inside. So our plants consisted of beans and uh, radishes, and the worms inside helped the plants increase growth through tunneling air and water that made it flow faster. And on our second layer, we have the soil, or sorry, the sand. So the sand makes the water flow into it and filter better into the aquatic chamber. And then our final layer, the aquatic chamber, where our pet sushi lives, along with one singular iglonia plant. So the group members that helped me with the project is me, Benny Chen, Jennifer Agner, Griffin Krog, and Stanley Chen. Alright, let's dive in. So since Sushi is our main character, we obviously have to do the nitrate level of the aquatic chamber first. The nitrate and nitrite had an overall increase as the ecocolumn lasted longer. The main reason why it increases is most likely due to Sushi pooping into the water and washing the soil's nitrogen into the water too. So Sushi eating and pooping is normal, like all organisms do. So because sushi has to live in its own poop, the nitrogen level also increases. And the soil nitrogen also gets washed into the aquatic chamber, but which most likely has minimal impact. Okay, here's the graph for the nitrate and nitrite level. We didn't measure anything prior to day 7 because sushi was not in it yet. But on day 7, we inputted sushi and it started taking measurements. So as you can see, nitrate and nitrite were fairly low during the day, but as the days increases, so does the nitrate and nitrite level. This is most likely due to sushi continue to eating and pooping, and it increases the nitrate and nitrite levels. All right. So for the next slide, we have Stanley Chen filling in for me.
Hello, this is Stanley Chen here, and I'm here to talk about the soil measurement. The soil data was almost exactly the same throughout the whole experiment. The fertility rate was low compared to normal fertility. It was like 1 to 3 throughout the experiment. As you can see, the temperature was almost throughout a 70, and the pH was almost like a 7 or 6 throughout the whole experiment. The fertility was the lowest one, as you can see, and it was like a 2 or 3, or maybe even a 1 sometimes. So how did the other eco-columns affect our? The other eco-columns were put in the same spot as our eco-column, and they received the same light that we did. They also had fishes in their eco-column, which were like our fish sushi, and the sushi had the worms above them, which we put 2 of them in for our sushi's neighbors. We also thought that there was some sort of disease passing around throughout the eco-columns because all the leaves are turning a yellowish color. When our leaves turned yellowish, we took off the leaves and buried in our soil for the nutrition. When we watered the eco-column too, the sushi also got the nutrition throughout the soil because when the soil water gets passed down into sushi's water. Okay, next up we have Jennifer Agner.
Okay, so I'm Jennifer, and for our observations, we'd say the bean plants grew the most and at a very fast rate, especially compared to the other plants, which barely grew throughout the entirety of the ecocolumn. For our water, the more days it lasted, the browner it got, which is most likely due to filtration of nitrogen and other nutrients filtering down into the aquatic chamber, and also because of sushi's waste and bacteria, anything coming off on it throughout the process. And at the end of this assignment, Sushi does not want to live up to his name. He does not want to be put in somebody's mouth or ingested. He wants to actually pursue freedom. And for questions to ponder, our first one is, to what extent did the detritivores affect soil nutrition and fertility? Which is basically, after adding the worms into our ecocolumns, like how was the pH affected, how was the temperature affected, how was the fertility affected. For our second one, we said, what effect do the conditions of the aquatic organism have on its ecological tolerance? So basically, how did the different conditions, how did the different levels of stress affect our organism, and like to what extent would that be? For our third, we have identify the extent of the stressful conditions the aquatic organism was able to tolerate. So basically, how stressful our conditions of the ecocolumn got and what it was basically able to endure. We have, since there was limited space for production, how did the plants affect one another and to what extent? So basically, like if one plant were to grow like larger, or if it could produce more beans, for example. And our last one is, what amount of oxygen and glucose was produced through the process of cellular respiration and photosynthesis, and what effects did it have on the aquatic chambers? So basically, we would have to pay close attention to the production of the plants, or the growth of the plants, and then the conditions of the aquatic chamber.